In recent years, scientists have really begun to look at individual cells and functions of individual cells. The 10x Chromium controller allows scientists to do gene expression studies in individual cells instead of an entire pool of many different cells. A good way to look at single-cell sequencing is by thinking of a smoothie versus a bowl of fruits. Whole tissue sequencing, where there's millions of cells, millions of different types of cells, it's kind of like a smoothie. You've got many fruits in that smoothie. And when you take a sip of that smoothie, you get a nice, creamy, fruity taste. But when you eat a bowl of fruits with each individual fruit separately, you then get to actually get the taste of the strawberry or the raspberry. So this is kind of how single cell is. When you look at a whole group of cells, you know that the genes are there, but you don't know, is it the tumor cells or the T cells or the B cells that are expressing those genes? But when you do single cell sequencing, you know exactly which cell is expressing those genes. Scientists then can use that information to do targeted therapy and to fight diseases. The machine uses microfluidics. So you know how when you mix water with oil, you shake it up, you get this emulsion. And the emulsion are like teeny little bubbles. So basically that's what's going on in the 10x Chromium. You're getting teeny little bubbles, just big enough to fit one single cell. And that's how you partition each cell. So in those millions of bubbles that are in the emulsion is one single cell per bubble.